Hi everyone, and welcome to this lecture. In this lecture, we're going to be covering Chapter 15 of your textbook, covering intracellular compartments and protein transport. The textbook does go into some depth around some of the aspects of this topic, so again, you just need to know this topic at the level that we're going to cover in this lecture. All right, so the key concepts for Chapter 15 that we're going to cover, we need to be able to describe the mechanisms by which synthesized proteins are then transported around eukaryotic cells. And we want to be able to understand and explain the basic principles and functions of exocytosis and endocytosis. So there's 5 areas we're going to be covering in this lecture. We'll start by just reminding ourselves about membrane-enclosed organelles or membrane-bound organelles. We'll then look at protein sorting, and then we'll go on to talk about vesicular transport and secretory and endocytic pathways. So first up, our membrane-bound organelles. We already know that eukaryotic cells have membrane-bound organelles, and they're bound by at least one membrane, and in some cases, 2 membranes. And what these organelles do is allow the compartmentalization of different processes and of different molecules. We can separate our processes, and in that way, it gives us greater control of some of these processes. So we can see in this table the main cellular organelles and the functions of them. And we'll go into detail into some of these in other chapters, but today the ones we're particularly going to concentrate are these: the endoplasmic reticulum, the Golgi apparatus, lysozymes, and endosomes. So first up, we're just going to go through each of those organelles and remind ourselves of their basic structure and function. So first up, the nucleus. We're very familiar with this. It's surrounded by our double membrane, our nuclear envelope. And there are nuclear pores that are embedded into this membrane that allow movement of molecules into and out of the nucleus. And we'll talk about that more in a moment. The important thing is this outer membrane is continuous with the membrane of the endoplasmic reticulum, and we'll talk about that now. So our endoplasmic reticulum, it's a series of membranous sacs and tubes, and as we said, continuous with the nuclear envelope. There's 2 different types of endoplasmic reticulum: rough endoplasmic reticulum, which has ribosomes embedded in that membrane, and smooth endoplasmic reticulum, which lacks ribosomes. There's a number of different functions of this organelle. It's very important, as we saw, in the synthesis of new membranes. It's important in protein synthesis, obviously, in the rough ER with the ribosomes attached. But it's also involved in the synthesis of other molecules, including things like hormones. The Golgi apparatus is another membrane-enclosed organelle. What it does is receive proteins and lipids from the endoplasmic reticulum. It then carries out a variety of potential different modifications to those proteins and lipids, and then it sends them on to their next cellular destination. The other types of organelles we'll be talking about later in this lecture include lysosomes, and these carry out a variety of degradative functions. Peroxisomes, which also carry out degradative functions, particularly around lipids and toxic molecules. And then we'll also take a closer look at transport vesicles. So how did these membrane-bound organelles evolve? We know that prokaryotic cells lack these membrane-bound organelles, so how did these evolve? Well, it's thought there's 2 different mechanisms. The first is that originally our DNA of our ancestral cell would have been attached to the membrane, and so were the membrane-bound ribosomes. It's thought that what happened probably was that some of that membrane then invaginated or turned in on itself and then enclosed that DNA. And then extending from that was further membrane, which had the ribosomes attached to it. We saw earlier in semester about how mitochondria were formed. So mitochondria were formed when one of those ancestral cell with that newly formed nucleus engulfed a bacteria that was capable of aerobic respiration. And then so the mitochondria were formed with a double membrane around them with an origin from that aerobically respiring bacteria. And we also saw how chloroplasts were also formed in a similar way to this, the engulfing of a photosynthetic bacteria. So there's two quite distinct mechanisms there of formation of these organelles. So we know that the membranes of many organelles in eukaryotic cells are either connected to each other directly or can readily fuse with each other. But some of them don't. And this is connected to the fact of these different origins. So the ones that are either connected or can readily fuse are the ones that were generated in that first mechanism by invagination of the plasma membrane. So the ER, the Golgi, and the plasma membrane itself, for instance. The ones that don't, well, they're our mitochondria and our chloroplasts because they were generated by this second mechanism, which is a completely different origin. So what we're going to look at now is how once proteins are synthesized, how they're directed to the correct compartment or the correct organelle that they need to be in for their function. Now we know that proteins are largely synthesized in the cytosol at the ribosomes that are present there, and the ribosomes can either be free in the cytosol or they can be attached to the endoplasmic reticulum membrane. I say largely because there are other places that protein synthesis occurs in the cell. We know that mitochondria and chloroplasts have a bacterial origin. They have their own DNA and they are capable of synthesizing a small amount of proteins themselves. But what we're talking about for these examples is those proteins that are made by the ribosomes in the cytosol. So once they're synthesized, there's a number of different fates they can have. Their role might be to remain in the cytosol, and in this case, they lack what we call a signal sequence. So what these signal sequences are is essentially postcodes for the proteins that tell the cell where they should deliver that protein to. So if they have one of these signal sequences, they can be transported around the cell by a number of different possible mechanisms. The first possible mechanism is they might be delivered to the nucleus and they will enter the nucleus through nuclear pores. Another possible way they can be transported is they can be transported across the hydrophobic membrane of an organelle in an unfolded state. And this largely happens in mitochondria and chloroplasts. The other way they can be transported is by vesicles, and this allows them to be transported to other organelles or even exported from the cell. And this process involves fusion of membranes of organelles and vesicles and potentially the plasma membrane. So we can see here examples of those signal sequences that are specific for the target of that protein, their cellular location. So what they are is short sequences of amino acids that are at the end terminus of the protein, so the start of the protein. And they can be characteristic for different locations that they're directing the protein to. So for instance, if we look at import to the nucleus, we see these signal sequences involve a string of positively charged amino acids. And for import into the endoplasmic reticulum, a string of hydrophobic amino acids. So while they can be slightly different on different proteins, the final destination of the protein, each of those signal sequence has a characteristic format. So now we're going to look at each of those three possible mechanisms in a little bit more detail. So the first one, transport into the nucleus through nuclear pores. So we can see again here the characteristic signal sequence with a string of positively charged amino acids. Now the textbook goes into some detail around this process, but we only need to know it at this level. So the nuclear pore is a large complex and consists of around about 30 protein molecules. So it's a large complex molecule. It is selective in that it does allow small water-soluble molecules to pass freely, but these larger molecules, such as proteins, need an appropriate signal to be able to pass through the pore. Transport across or through that nuclear pore is powered by hydrolysis of GTP. So again, storage of that energy in a high-energy phosphate bond. The second mechanism we're going to talk about is when proteins are translocated across membranes in an unfolded state. And this tends to happen with proteins going across into the mitochondria and chloroplasts. So the signal sequence directs them to the outer membrane, and then the protein will pass through the outer membrane and then through the inner membrane through a translocator. Once the protein is inside that organelle, the signal peptide or the signal sequence is cleaved from the beginning of that protein, and the protein is then folded into its mature conformation. This process, once inside the cell, involves chaperone proteins, and that process is powered by hydrolysis of ATP. So proteins which are destined to be delivered from the cytosol to a peroxisome, most of them also get transported in this way. So the signal sequence on these proteins is recognised by a receptor protein in the cytosol. One or more of these receptor proteins, but at least one of them, takes the protein to the peroxisome, translocates it across the membrane, and then returns back to the cytosol. Again, this process involves chaperone proteins and is powered by the hydrolysis of ATP. There is another possible way for proteins to be delivered into peroxisomes, which we'll talk about shortly, and this is when they're delivered by a vesicular transport. The last mechanism involves proteins which are synthesized in the cytosol being delivered to the endoplasmic reticulum. We know that our ribosomes can either be free in the cytoplasm or they can be attached to the endoplasmic reticulum membrane. And the reason why they get attached to that membrane in some cases is that a signal sequence, which is at the start of that growing translated protein, directs that ribosome complex to the ER membrane. Then as that protein is being synthesized in the ribosome, it is physically pushed through a protein translocator embedded in the membrane and into the ER. So we can see here at the top an example of the type of signal sequence that does that process, that directs that newly synthesized protein to take the ribosomal complex to the endoplasmic reticulum membrane. Once they're being synthesized and physically pushed through the transporter across the endoplasmic reticulum membrane, there's two possible things that can happen to that protein. They can go directly across the membrane and into the endoplasmic reticulum lumen. This tends to be the case for water-soluble proteins, and these proteins are either destined to remain in the lumen of the endoplasmic reticulum or be transported to the lumen of another organelle. The other possible thing is they might be embedded in the endoplasmic reticulum membrane, and this tends to be the case for transmembrane proteins. So these are proteins that their ultimate destination is to be embedded in a membrane, either in another organelle or indeed in the plasma membrane. So in both these cases, if their final destination is not the endoplasmic reticulum, they are then transported to that final destination using vesicular transport. So what is it that we mean by vesicular transport? Well, this kind of transport involves the use of membrane-bound vesicles, and the membranes of these vesicles are capable of fusing with other membranes. So fusing with the membrane of the endoplasmic reticulum, of the Golgi, of lysosomes, and indeed with the plasma membrane itself. So what this is able to do is not only link organelles by allowing their membranes to fuse, but also it can link the interior of the cell with the external environment. And the process where this happens, these two processes are known as exocytosis, when cellular contents are released to the environment, and endocytosis, when external molecules are taken up by the cell. So this allows our cell to import and export molecules. So this is an overview of how the process works. So we have on the far left our endoplasmic reticulum. What happens is parts of the endoplasmic reticulum membrane bud off into these membrane-bound transport vesicles. These then are transported to the Golgi apparatus, and through a series of processes through the Golgi apparatus that we'll talk about a little bit in a moment, they can then be passed out the other side, or the trans side of the Golgi apparatus network, and into transport vesicles. If these vesicles are transported directly to the plasma membrane and fuse with the plasma membrane, they can release their contents to the external environment of the cell, and that's known as exocytosis. Another option is that some of these transport vesicles could then go to an endosome, and we'll talk about that as well. So endocytosis is the process whereby the membrane can bud in, bringing molecules with it. This forms into an endosome and then fuses with a lysosome, and we'll talk about that more in a moment. These are highly organised processes. So the vesicles fuse with the membrane at the other location, and the key thing about this is they need to only take with them the appropriate proteins or other molecules that they're supposed to have with them. So it's a very organised process. Now, we don't need to know the details of how this process works, but the process by which those vesicles are transported around is really complex. So the cytoskeleton, the proteins involved in that, can assist with the movement of the vesicles, and then there's a series of other molecules that help the vesicle to dock and then to fuse with the membrane. So a very complex and organised process. So now we'll move on, and firstly we'll talk about exocytosis, about those secretory pathways, and then we'll finish up by looking at endocytosis. So as we saw, our proteins have made their way into the endoplasmic reticulum. And once they're there, either in the lumen of the endoplasmic reticulum or embedded in the membrane as we saw, these proteins can be covalently modified. Some of the common modifications that happen include disulfide bonds. So we've seen this earlier when we looked at protein structure, when covalent bonds form between sulfur atoms in two cysteine residues. Now, as we saw then, these don't normally happen in the cytoplasm due to the reducing conditions that are present there, but it's possible for this to happen once in the endoplasmic reticulum. And the role of these is to provide extra strength to protein structure because they're covalent bonds, very strong bonds. Another modification that can occur is glycosylation. This is when a carbohydrate molecule is added to the protein. And these carbohydrate molecules are pre-synthesized. They consist of 14 sugar monomers, and they're added at specific recognition sites in the protein sequence. So there's a specific recognition site of 3 amino acids, and the amino acid that the molecule gets added to is an asparagine residue. These carbohydrate molecules can be further modified, as we'll see in the Golgi apparatus. So a key thing that happens before proteins are exported from the endoplasmic reticulum in a transport vesicle is that they are thoroughly checked for errors. So they're checked to make sure that they have correctly folded, and if they assemble into larger, more complex structures with other protein molecules, such as happens in antibodies, they're checked to make sure that assembly has occurred correctly. This process is assisted by chaperone proteins, and any proteins which fail these checks are going to be exported from the endoplasmic reticulum back to the cytosol where they're degraded. Once the proteins pass this checking in the endoplasmic reticulum, they are budded off into transport vesicles, membrane-bound vesicles, and transported to the Golgi network. Now, the Golgi network is a really complex structure. We can see here on the left, it has a series of folding membranes. The ER-facing side of the Golgi network is known as the cis Golgi network. It gradually works through the cisterna, the cis medial and trans cisterna, until it gets to the trans-Golgi network, which is on plasma membrane facing side of the Golgi network. So proteins progress through this Golgi network from that cis-Golgi right through to the trans-Golgi. And they can do this by one of two different mechanisms. They can again use transport vesicles. So the vesicles that come from the endoplasmic reticulum will fuse into the Golgi network, and as they progress from one part to the next, they will butt off into transport vesicles. The other way that they can be transported through the Golgi network is that actual parts of the Golgi membrane itself will fuse with the next part of the Golgi. So the membrane is sort of transported through the Golgi network. Further modification of any carbohydrates that were added in the endoplasmic reticulum can happen in the Golgi. And once those processes have happened, our proteins are ready for the next step in the transport pathway. Once our proteins have progressed through the Golgi apparatus and have butted off into transport vesicles from the trans-Golgi network, there's two possible pathways to enable them to fuse with the plasma membrane. The first is known as the constitutive pathway. This pathway operates continuously. A couple of the key things it does is not only can any soluble proteins be packaged up and then transported to the membrane and released to the external environment, it's also the mechanism cells use to deliver new lipids to the plasma membrane. So the transport vesicle buds off, makes its way to the plasma membrane, the two membranes fuse, and the contents, if there are contents, are secreted to the external side of the cell, or new lipids can be inserted into the plasma membrane. The other pathway is known as regulated exocytosis. In this mechanism, again, the proteins are packaged up into a transport vesicle at the trans-Golgi network, but in this case, they remain in a secretory vesicle, storing these secretory proteins. Once the cell receives an extracellular signal, this can be something such as a hormone, that triggers that secretory vesicle to then fuse with the plasma membrane and release its contents. So lastly, we're just going to talk about endocytic pathways. What these pathways do is essentially the opposite. They allow molecules to be brought into the cell. And the types of things that the cell regularly brings in are things like fluid, small soluble molecules, but also larger molecules and even whole cells. So what happens is a section of the plasma membrane is progressively enclosed until it forms its own vesicle. There's two quite different endocytic pathways. One's known as pinocytosis and the other's phagocytosis. So pinocytosis is known as cellular drinking. It's largely fluid and small molecules that are brought into the cell using this mechanism. Again, this mechanism happens continuously, and it's a random process that the plasma membrane will just bud in to form that vesicle, taking up fluid and water-soluble molecules that are in the vicinity. It can also happen via a selective process, and that's known as receptor-mediated endocytosis. And things like cholesterol, so molecules that aren't water-soluble, are taken up by this method. So they bind to a receptor on the surface of the cell, and that triggers a process of endocytosis. The other process is known as phagocytosis, and we can think of this as more cellular eating. It involves the uptake of large molecules, of pieces of cell debris, or even of whole cells in the cases of microorganisms. Generally, there are specialised cell types which carry out this type of function. So in this figure, we can see how these processes work once the molecules or cells have been taken up into vesicles. So in pinocytosis, the vesicles go through a series of endosomes as the endosome matures. So firstly an endosome, and then that endosome will lyse with a lysosome. In the case of phagocytosis, if we're engulfing an entire bacteria, again, it is surrounded by the plasma membrane until it forms its own little vesicle, and that's known as a phagosome. That again will then lyse with a lysosome. There's also a process known as autophagy, and that's when cellular debris within that cell is engulfed and broken down. So in some cases, we see this happen with mitochondria in particular. Mitochondria can then be surrounded by membrane and then will undergo that process where they're fused with a lysosome and degradation occurs. So what happens inside lysosomes? Well, they have an extremely acidic environment, and the acidic environment is maintained by an ATP-driven proton pump that pumps protons into that lysosome to maintain that lower pH. The lysosome also contains a whole range of different degradative enzymes. These enzymes are synthesized in the endoplasmic reticulum, transported to the Golgi, and when they're in the Golgi network, they're tagged with a particular phosphorylated sugar group. So instead of them being exported via exocytosis, those tagged proteins are sent to lysosomes via endosomes. Once our molecules have been delivered to the lysosome for degradation, all of these enzymes can then play a role in the degradation of different molecules. Once that degradation has occurred, the individual building blocks of those degraded molecules can be recycled. Whole molecules may be even recycled out to the rest of the cell. So a very efficient process here. And we're keeping all that degradative work contained within that organelle so it doesn't affect the contents of the rest of the cell. So that's the processes by which our synthesized proteins are able to be transported to the various parts of the cell by these really targeted and well-controlled processes. And it's really important also the way that cells are able to transport proteins out via exocytosis and bring things in that they need via endocytosis. So that covers our protein sorting and vesicular transport at the level we need to know it. So we've described the mechanisms by which those synthesized proteins are transported, and we've discussed the principles and functions of both exocytosis and endocytosis. So thanks everyone, that was Chapter 15.